Hey everyone, this is Elad from Astrolabe Diagnostics, and in this video I'm going to describe how to set up a new experiment in Astrolabe which uses differential expression analysis. For the purpose of this demo, I am going to use a publicly available dataset that comes from Glassberg et al. And in this study, which involves patients that have sickle cell disease, the authors applied an extracorporeal perfusion system to model the effects of cells trafficking through blood vessels, and we would like to examine changes in marker expression before and after the application of this system in both healthy individuals and patients with sickle cell disease. I am going to briefly go through the feature setup for this experiment. Please note we have a much more extensive tutorial just on this subject, which I recommend that you go and check out. So I'm going to add a few features that will help us navigate through this data. First of all, the different patient IDs. So we have three control patients and two sickle cell patients. And I'm going to enter these identifiers, which Astrolabe will automatically match to the file names. I am going to also add condition, which can be either pre or post application of the system. And finally, I'm going to add a type point, which can be 10 minutes, 30 minutes, or 60 minutes. All of these have been automatically matched to the file name. I'm just going to briefly review them to make sure that this is the case. Next, I would like to do some more sophisticated setup. So for that, I'm actually going to download all of the features as a spreadsheet and continue this work in Excel. The first thing is I'm going to change the sample names to something which is a bit more edible. And we're going to change the sample names to the format patient, time point, condition. Then we are going to add the patient group. And we have two groups. We have patient and we have controls. So we're going to set up all the patients and we're going to set up all the controls. Then I would like to add three auxiliary columns that will allow us to compare every time point separately. I'm going to call them time point 10, 30, and 60 respectively. And in each one of them, I would like to examine the cross between the patient and the patient group and the condition. So we're going to once again concatenate, sorry about that, and we're going to concatenate the group and the condition. So we're going to do this, and just to make things a bit more comfortable to look at, I am going to replace all of the instances of patient with P and all of the patients of all instances of control with C. And let's copy these here. We don't need this one anymore. Now at this point, this will compare all the different time points. We would like to compare one time point at a time. So I'm going to sort the spreadsheet by time point, 10, 30, 60, and every value that doesn't fit a given column, I'm going to set to an A. So this is the 10 minute, so only the 10-minute samples. This is the 30-minute, so only the 30-minute samples. And this is the 60-minute, so only the 60-minute samples are going to be set. Now that we have this spreadsheet in place, we are going to go back into Astrolabe and upload this template. And of course I forgot to save it as a CSV, so let's try again. And now the import worked, and we can see all the different time points, all the information we put in inside the platform. Now, one last thing I want to do here is change the order of the feature values. And what do I mean by that? I'm going to click on this title time point 10, and we see that there's 4 different values here: C post, C pre, P post, P pre. And this is the order where the values will appear throughout the platform. And I actually want cpre to appear before of cpost and then ppre to appear ahead of ppost. And I'm going to make the same change here, and finally I will make the same change here. We're now ready for the differential expression analysis itself. I will click on the Analysis tab and on Add Differential Expression Analysis. There's a few parameters that we need to set here. The first one is going to be primary feature, and I'm going to pick time point 10. As a reminder, it's going to compare the control and the patients before and after treatment at the 10-minute time point. You will notice that the name is being populated automatically. I can change it if I want to something else. However, I'm going to leave it at time point underscore 10. Then we need to set dependent features, and this will be incorporated into the statistical model, for example, for the purpose of pairwise statistics. So I'm going to add patients, since we have pre and post for each patient. This is pairwise analysis. The next thing I'm going to discuss is choosing the markers for the analysis. And over here, you can include any marker which you conceivably expect to change. Now, just for your own comfort and also for multiple testing correction reasons, I strongly suggest that you stick to those markers that you actually think are going to change across the conditions. So, for example, I am going to skip surface markers such as CD56, CD19, CD3, which I know are being used as part of immunophenotyping. Then, there's two more options here that I would like to discuss. The first one is marker statistics, and this indicates which aggregate statistic we're going to use to calculate the intensity values that we would like to compare. Astrolabe defaults to the 95th quantile. This is the most sensitive way. We focus on the long tail of the distribution, and this is what we want to compare. If you would like to be slightly less sensitive, you can click on median and use that instead. Please note we have a separate video talking about differential expression analysis and explaining these differences. And the last thing you can choose is a reference feature. A reference feature would be used as a baseline, as a method of calculating background intensities. In the case of this experiment, we don't really have one. If we would have wanted, we could have used a condition for it. In this case, we would use pre as a baseline, and this means that all of the pre time points would effectively be 0, and for every patient we would look at post versus pre in our modeling. However, we would like to keep the pre value so we can compare controls versus patients. If this was just controls or just patients, we might have used this as a reference. A few other common examples for a reference include reference spike-ins, if you would like to use these, or unstimulated conditions. Let's say you have 2 or 3 different stimulations that you would like to compare. In all cases, the unstimulated is the baseline. Though again, in this case, we're just going to leave it blank. And I'm going to create the analysis. Now, this analysis is only for one primary feature, time point 10. However, we have 3 different time points. So I'm going to click on this clone button to create 2 more copies. And then I'm going to change them to time point 30. And you will notice that everything remains the same except for the title. And I'm going to do the same for time point 60. Now the analysis is done, it's ready. We're almost ready to begin. One last thing I will mention, always check your channels tab. In the case of this experiment, there might be a few changes we would like to make. So I'm going to take a quick look, and it seems to be fine. However, please review the experiment setup tutorial to see if you should change anything in your channels tab. So that's it, we're ready to start the experiment. I'm going to click on continue and I'm going to confirm. And as soon as I do this, Astrolabe's analysis is going to begin. And please join me in the next tutorial where I'm actually going to skip the actual analysis. It's going to take about an hour for the platform to crunch through all this data, and within an hour you can access the results. And please join me in the next tutorial on how to analyze the results of the analysis and how to go through the different visualization and figures that Astrolabe offers. And with this, I will thank you for your time. If you have any questions, please email me at elad@astrolabediagnostics.com, or at support@astrolabediagnostics.com. Also, please remember to subscribe to us on LinkedIn and on YouTube to get all the latest tutorials and videos. I'll see you in the next video where I talk about the results.